welcome to this webinar, Developing Gene Therapies for Cardiomyopathies, jointly sponsored by both Hart University and the SADS Foundation, or the Sudden Arrhythmia Death Syndromes Foundation. I'm Marcia Baker, the Program Director with the SADS Foundation. So on behalf of both Hart University and SADS Foundation, it's my pleasure to introduce our speakers today. Our presenter is Dr. Andrew Landstrom. He is an associate professor of pediatrics at Duke University School of Medicine. He's a physician scientist and cross-trained in pediatric cardiology, electrophysiology, human genetics, and molecular physiology. Dr. Landstrom is also a member of the SADS Foundation's Board of Directors. Dr. Nicole Weaver is a cardiac geneticist at Cincinnati Children's Hospital and will be moderating the Q&A portion following Dr. Landstrom's presentation. So thank you both for your time. And now I'll turn the microphone over to Dr. Landstrom.
Thank you very much, Marcia, and thank you all for joining us to talk about developing gene therapies for cardiomyopathies. I have no disclosures, conflicts of interest, or relationships with industry, although I do accept research and salary support from a number of not-for-profit and federal organizations. So our three objectives for our CME time today are to detail the advances that have made gene therapy possible, to illustrate early phase clinical trials for gene therapy, specifically in cardiomyopathies, and to illustrate potential hurdles in bringing gene therapy to the clinic. When we talk about cardiomyopathies, this will probably be review for many on the call, but cardiomyopathies are really a phenotypic spectrum of disease, all defined by a commonality of being a primary disease of the heart muscle. There are many different forms of cardiomyopathy, all with different phenotypes. For example, there's hypertrophic cardiomyopathy, defined as asymmetric hypertrophy of the left ventricle in the absence of causes that can trigger the heart to hypertrophy. There's non-compaction cardiomyopathy, where the heart muscle fails to form and compact normally in development, which sort of underlies an abnormality that persists after the patient is born. Restrictive cardiomyopathy, where the functional defect is not the contraction of the heart, but the relaxation of the heart muscle, leading to high end diastolic pressures and restrictive physiology. There's dilated cardiomyopathy, where the left ventricle dilates abnormally and loses systolic function. And arrhythmic cardiomyopathy, where the heart muscle is actually replaced by a mixture of fat and of scar, which can be highly arrhythmogenic and can result in heart failure. And so all of these are different types of cardiomyopathy, and all of these have at their central core that they are actually genetic and thus heritable. This is the kind of genetic result oftentimes that can be expected from patients with cardiomyopathy. This is a pathogenic positive genetic test for a variant in MYBPC3, which is one of the most common causes of hypertrophic cardiomyopathy. And if you kind of delve into this genetic test result a little bit, you can see that it is a loss of function variant with a sort of insertion that shifts the open reading frame of the gene, resulting in early truncation of the protein, leading to a loss of one of the alleles, loss of the protein expression of one of the alleles. And it's heterozygously, and the known mechanism of NYBPC3 mediated disease is that of haploinsufficiency. And so this sort of open and shut genetic diagnostic test is really made possible by a clear understanding of the genetic underpinnings of disease, and really highlights the importance of genetic testing for all patients with suspicion for cardiomyopathic disease. And when you apply gene testing to patients with cardiomyopathy, you'll see that all types of cardiomyopathy have a genetic origin, but the yield of this gene test is a little bit different based on the phenotype of the patient. So for example, we know a lot about the genetic causes of hypertrophic cardiomyopathy and arrhythmic cardiomyopathy, so much so that if you see these patients, that gene test will be positive 70 to 80% of the time. Conversely, we don't know all the genetic causes of things like dilated cardiomyopathy or non-compaction cardiomyopathy, and thus the genetic yield there is lower. But the thing to remember here, regardless of the yield, is actually that all of these are genetic. Thus, understanding the genetic etiology of these diseases is key, especially given the wave of gene therapies that we are seeing kind of break upon clinical trials and to come to fruition, we hope, in the clinic. And so to take a step back, I define gene therapy as anything that is a genetic modification of a cell that produces a therapeutic effect. And historically, gene therapy has sort of a troubled origin, dating all the way back to sort of the 1980s and 1990s. This is Ashanti De Silva, who was actually the first patient who was successfully treated with gene therapy. She didn't have cardiomyopathy, didn't have a cardiac disease. She actually had severe combined immune deficiency. And in 1989, Steven Rosenberg at NCI successfully treated her with a retrovirus that dropped in a gene into the white blood cells that were deficient, leading to SCID. And because of that, she actually reconstituted the molecular defect behind her disease, had a relatively normal immune system, and actually leads a relatively normal life. In fact, she's actually now a genetic counselor, which I think is actually really quite remarkable. However, just as this success really benchmarked the possibility of gene therapy in the clinic, very early on, there were long-term complications. And so this is a paper from Science in 2003 that looked backwards in time and actually found that a lot of the patients who received that very same gene therapy for SCID actually had a predisposition for oncogenesis, because the area in which the gene inserted that was used in this gene therapy was sort of unconstrained and led to the development of cancer in a small number of patients. And then, of course, in 1999, there was the death of Jesse Gelsinger. Jesse had ornithine transcarbamylase deficiency, and was given an adenovirus with a corrected gene to hopefully resolve his metabolic disease. But he had a dramatic immune response to the virus and actually ended up dying. And so scientists were really sort of kind of put on the ropes and asked, why is it that this happened? How could somebody actually die of gene therapy? And there was a lot of retrospective evaluation of what exactly happened. And questions were raised about whether actually Jesse was too sick, perhaps, to undergo gene therapy. Perhaps he and his family did not know the full extent of the possible side effects, including death, at this sort of early stage in gene therapy. Perhaps the university didn't fully disclose the side effects in time. And so this raised a lot of red flags very early on. And there was actually a moratorium that was placed on gene therapy following Jesse's death and some other missteps that occurred very early. And so the challenges back in the 1990s were many. There were challenges around how DNA would be delivered, and challenges around ensuring that there were no off-target effects, such that the DNA targeting that did occur would occur only in one place and not somewhere else in the genome. Back then, and I would argue to some degree now, gene therapy was not tissue specific, meaning you might try to target one organ, but you might get an entirely different organ, leading to increased risk of side effects and potentially mortality. There is, and there still is, a lot of concern around the reaction of the immune system to the virus, specifically to the viruses that we use for gene therapy, particularly for the heart. And this is a major hurdle that must be sort of gotten over for safe gene therapy delivery. And then back then, the only way to actually pursue gene therapy was to replace a gene. That is, somebody had a pretty significant gene defect that wasn't functional, and that gene was simply introduced into the cells that were deficient. However, that would actually leave out many, many patients with many cardiac diseases who don't have a pure loss of function genetic mechanism. And so because of this, there was nearly a 2-decade moratorium on gene therapy. The field did not really progress. However, in the past few years, this moratorium has really been lifted, and it's been driven by a number of scientific advances that I believe have led to the resurgence of gene therapy, and more broadly represent a catalyst for an oncoming wave of genomic medicine in cardiology. The first such advance was the precipitous fall in the cost in sequencing the human genome. So shown here is an often shown curve. On the y-axis in a log 10 scale is the cost of sequencing a single human genome. And on the x-axis is time. And so way back in 2001, it cost about $100 million to to sequence a human genome. And now in 2024 at Duke, we can sequence a human genome for about $400. And so this precipitous decline, known as Carlson's curve, was incredibly rapid. And just for reference, this is Moore's law in the white line, which is something like, the speed of computer resistors doubles every 18 months. The cost of genome sequencing fell faster than the processing power of computers actually increased. So this is one of the advances that actually has driven forward genomic medicine and gene therapy, the cost of genome sequencing. A second major innovation was the discovery of CRISPR-Cas9. CRISPR-Cas9, for those of you who may not know its origin story, is actually an immune molecule carried by bacteria that bacteria use to prevent infections from viruses. And what it basically is a pair of molecular scissors that can be targeted very specifically to known areas of the human genome. And that can lead to a very precise cutting of the DNA, which then allows for things to be inserted. And they can be small things or they can be big things, but inserted very precisely into the human genome. And so this has opened up a world of opportunity for gene editing and for gene therapy more broadly. Another major advancement are these. These are cardiac myocytes that are made from induced pluripotent stem cells. And so individuals can have their blood drawn, their stem cells derived from their white blood cells, and then these stem cells guided to become really any cell in the body. I mean, you can imagine how being able to guide these iPSCs into cardiac myocytes allows for a very important model to be able to learn more about genetic disease, but also to test new therapies in human tissue, in potentially diseased human tissue, before bringing them to clinical trial. Another major advancement is the recognition that some viruses and some viral subtypes are actually very good at infecting certain organs. And in particular, AAV9 is actually really good for cardiac gene therapy. It wants to go to the heart. It wants to deliver payloads into cardiac myocytes. And the recognition that this could be done is actually a major step forward. As we discussed, it actually brings about a number of other concerns, as, for example, the immune system's response to this virus. But in theory, AAV9 can be actually a very high fidelity way of conveying sort of genetic information into the myocardium in a highly efficient manner, and a major advancement that's allowed for gene therapy to progress into clinical trials. And the lipid nanoparticles are actually something kind of on the horizon. We talked about sort of the immune response to viruses being a major cause of the morbidity and mortality associated with gene therapy. In theory, that would be greatly diminished when you're not using a virus. Something like lipid nanoparticles can be made to actually encapsulate DNA cargo and RNA cargo that can be safely delivered to cells. This is actually the foundation of the RNA vaccines for COVID that made them so successful in their delivery. And there's a lot of work being done in actually using these nanoparticles for gene therapy. So something on the horizon. There are many potential ways to deliver gene therapy. Naked DNA, RNA, or protein is actually a way that could potentially be used for gene therapy. We talked a little bit about sort of nanoparticles and microsomes and these lipid bilayers that can carry DNA messages and DNA gene therapies into cells. And then a number of viruses, of course, that can be used. There's lentivirus, adenovirus, and AAV9. And among the viruses, AAV9 actually has very good cardiac tissue targeting. Among the viruses it's pure, has sort of a reduced propensity for triggering the immune system, thus reduced inflammation as one of its strengths. And its gene cargo can be expressed indefinitely. And so really the limitations for AAV9 are based purely on size. It can only take a certain size of a genomic insert. Potentially there are still some concerns for immune targeting of the virus, but overall it has a lot of strengths compared to other viruses, and it's highly efficacious. And so AAV9 has actually driven a lot of the gene therapies going forward, specifically for cardiomyopathies in the heart. And so how does the field look right now for gene therapies for various forms of cardiomyopathy? Let's start with hypertrophic cardiomyopathy, asymmetric left ventricular hypertrophy. So as many may know, hypertrophic cardiomyopathy is due to mutations in the proteins that encode these things, the cardiac sarcomeres, the elementary contractile units of the cardiac myocyte. Zooming into that cardiac sarcomere, there are many components that comprise the cardiac sarcomere, things like um, beta myosin heavy chain, alpha myosin, troponin complexes, titin, and actin. All of these are needed for successful myofilament contraction. And mutations in these genes that encode these constituent proteins are the cause of hypertrophic cardiomyopathy, as well as other forms of cardiomyopathy. And so understanding these genetic mutations has actually led to the ability to consider how we might actually attenuate that defect in the gene therapy space. And so, for example, mutations in MYBPC3, that example we saw way early as our genetic testing example, those are loss of function variants. And so thus, in theory, it would be quite possible then, if they're loss of function variants, to simply replace native MYBPC3 myosin binding protein C3 in cardiac cells, potentially as a gene therapy for hypertrophic cardiomyopathy. And that's actually the case. So right now, Tenaya Therapeutics has a MYBPC3 gene therapy replacement trial called MyPeak-1, currently in phase 1 clinical trials enrolling individuals with mutations in this gene and severe hypertrophic cardiomyopathy who are 18 years or older. Tenaya Therapeutics is not the only group, of course, with early phase clinical trials in cardiomyopathic disease. Rocket Pharma currently is enrolling for phase 2 clinical trials individuals with mutations in LAMP2, which is the cause of Danon disease, an X-linked recessive disease that can affect children, typically boys, very early on with very severe hypertrophy, among other manifestations of disease. And they're enrolling individuals out of UC San Diego and CHOP for phase 2 clinical trials, boys with mutations in LAMP2, 8 years or older. What about arrhythmic cardiomyopathy? What's the gene therapy space look like for arrhythmic cardiomyopathy? Well, ACM is actually defined by functional mutations in the genes that encode these things, the cardiac desmosomes. And so these cardiac desmosomes act like the rivets that hold heart cells together. And when the myofilaments contract, those desmosomes allow for those myofilaments to pull across the cardiac cell space, and so that the cardiac cells are linked together functionally, allowing for a myofibril to contract. And mutations in the components of these cardiac desmosomes lead to arrhythmic cardiomyopathy. And they come in many different varieties. For example, there's PKP2 mediated mutations in the protein plakophilin-2. Those are typically loss of function variants that result in haploinsufficiency of this key protein of the cardiac desmosome. And there are mutations, of course, in other components of the desmosome, including DSP encoded desmoplakin, desmogliin, desmocolin, and a few others. All of these are causes of arrhythmic cardiomyopathy when their constituent genes are mutated. Well, if we take, for example, PKP2 loss of function variants are the most common cause of arrhythmic cardiomyopathy. And is there an opportunity then to just replace the gene that's defective? Well, here are some of the preclinical trial work that went into the trials that followed. So this is a cardiac-specific inducible knockout mouse of PKP2. And when you knock out this gene, just like in humans, these mice develop arrhythmic cardiomyopathy. And you can see some of the primary data here of that model, which is actually a really good model for arrhythmic cardiomyopathy. You can see the knockout mice develop things like fibrosis, which is just like humans when they get arrhythmic cardiomyopathy. You can see that the RV by echo and by histology dilates. The LVEF decreases over time, and this results in actually mortality in these mice. Moreover, the mice get arrhythmias. They get ventricular arrhythmias, just like humans do. They get a high burden of inducible ventricular ectopy, and then you can actually trigger them to get ventricular arrhythmias, just like humans with arrhythmic cardiomyopathy. And so it's studies like this which identified the genetic cause of arrhythmic cardiomyopathy in a model that allowed for sort of in vivo validation of what a gene therapy approach might look like. And so the question became very simple. Could we take an AAV9 and load it up with PKP2, normal, healthy human PKP2, and put it into one of these mice? And these are the types of studies that resulted from that experiment. So this is from Tenaya Therapeutics, presented at the ASGCT annual meeting, annual meeting in 2022, showing in their hands what this mouse did when it received this therapy. In the black line is are the wild type mice who got no who were normal. In the blue line were the mice who got sort of a sham, and then in the red is a line of the mice that got the gene therapy. And as you can see, the ejection fraction of these mice dropped off when they got the sham, but when they got the gene therapy replacement, actually normalized to wild type mice. The mortality, which is actually quite marked in the sham mice who had the knockout, was recovered in the mice that got the gene therapy, very similar to that of wild type mice. And in addition, the arrhythmias went away. And so these are recordings of rhythm, ambulatory rhythm from the mice who had both the control or the gene therapy, identifying that the reintroduction of PKP2 in these mice resolved the arrhythmias that were being seen. Not only that, it actually showed that the phenotype could be reversed. Thus, if you actually allowed the mice to get sick and then delivered the gene therapy, one actually saw recovery of the RV dilation, recovery of the ejection fraction, and recovery of mortality in these mice. And this was all very exciting. And this is one study of a couple that demonstrated a similar beneficial phenotype when PKP2 was delivered to mice. And this, among others, was really the catalyst for a couple of early phase clinical trials in this space. And so Tenaya Therapeutics has the Ridge 1 study enrolling patients in a 1B clinical trial who were 18 years or older with a PKP2 mutation who were sick for PKP2 replacement gene therapy. Moreover, Rocket Pharma has one of these trials open as well, enrolling out of UC San Diego and CHOP, again, a phase 1 clinical trial, looking for individuals with PKP2 loss of function mutations who are 18 years or older. And so while the preclinical work and these early phase clinical trials are certainly exciting, there are certainly challenges ahead in the world of gene therapy. This is sort of a famous clinical trial in the gene therapy space that was a null trial. This is the CUPID-2 trial, which operated under the premise that one might be able to replace a specific protein, the sarcoplasmic reticulum calcium ATPase, or SERCA, in the setting of ischemic heart failure. So there's been a wealth of data over the years that individuals who get ischemic heart disease and ischemic heart failure have loss of functional SERCA, leading to a disruption of intracellular calcium in the cardiac myocytes. And that replacement of SERCA in these cells can be actually a way of rescuing some of the cell death and lack of contractility that occurs in heart failure. And so this group worked towards replacement of SERCA using AAV1, and the CUPID-2 trial was the phase 2B clinical trial of individuals who got AAV1 SERCA2 directly into their coronary arteries. And this is what they found. It actually really didn't work. And so their outcomes of heart failure related hospital admissions, the severity of heart failure, and then outcomes were no different among individuals who got a sham versus individuals who got the AAV directly into their coronaries. And we still don't really know the reason why. Could it be that there was no real efficacy in the virus, meaning the virus didn't actually transduce into the myocardium, or were there neutralizing antibodies in the individuals that actually prevented the virus from delivering its payload? We don't really know, but what we do know is that the trial showed no difference in its outcomes. So not only have there been challenges with consistently showing efficacy in clinical trials, there's also continued to be issues with mortality. So this is another somewhat famous case of a number of very late deaths in Astellas, which was previously Audentes, who was using AAV, particularly AAV8, for X-linked myotubular myopathy. And they notably had 4 deaths in individuals in their early phase clinical trials, and these deaths happened over a year after the virus was delivered. And there was some concern for perhaps the liver having failed in some of these individuals, particularly at the higher dose of the virus. But the fact that this could happen so late and so unexpectedly in 4 people is just really sort of, I think, probably illustrative of what some of the concerns are for gene therapy moving forward. Another famous case, this one, Terry Hogan, who was famously an N of 1 clinical trial, who was going to undergo CRISPR gene editing of his specific DMD mutation. He has Duchenne's muscular dystrophy, and he was going to have his mutation edited in his skeletal muscle to potentially regain some of his lost muscle strength. And he died. And it's actually unclear whether he even got the CRISPR-Cas9 delivered into his cells. Whether it was an immune reaction or not is sort of unclear. But again, one individual who had mortality because of gene therapy. And so despite the fact that back in the early 1990s, there was a moratorium with some early tragic deaths, now, if anything, there's just the opposite. Gene therapy still continues to be accelerating in the preclinical and clinical space, and we're seeing more and more gene therapies come to market. So what does the landscape of gene therapy look like now, in July of 2024? So these are a few statistics just to kind of give you an idea of what gene therapy looks like. And these are as of fiscal Q2 of 2024, so pretty up to date. There are currently 31 gene therapies approved by the FDA. There are 30 RNA therapies currently approved and 68 cell therapies that aren't sort of gene therapy, but cell therapies that have been approved by the FDA. The majority of the diseases that are being targeted with these approved therapies are cancer, skeletal myopathies, and metabolic diseases, and hematologic diseases. Cardiac disease is way down on this list. The pipeline is flush with potential targets. So as of right now, there are 4,047 gene, cell, and RNA therapies in preclinical trials all the way through pre-registration, and a little over 1,000 therapies specifically for rare disease, again, preclinical through pre-registration. 8 of the top 10 of these rare diseases are cancer, just to give you an idea of where people are seeing the most therapies being targeted. Rare diseases, though, are of increasing significance in this preclinical pipeline, and are oftentimes highly targeted because of the ability to make very specific gene changes in a well-defined mechanism. As you can see here, this is the number of therapies in the rare disease space over time. As you can see, there's many different delivery modalities, including RNA therapies. And RNA therapies are really exciting, coming on the horizon to offer another modality for potentially modifying cells. And as you can see, they come in many different varieties. Delivering straight up messenger RNA is the most common form of RNA therapy, but also interfering and antisense RNAs, things that are actually designed to suppress the expression of maladaptive RNA, mutated RNA, are actually increasingly in this space. There continues to be large investments in startup to be able to translate gene therapy from bench to the bedside. If anything, in the last few quarters, maybe a little bit of a decline in the total number of dollars raised for this kind of venture capital. And the total number of sort of mergers that are occurring may be declining over time. But if you look at the y-axis here, this is in millions of dollars, still tremendous investment in the preclinical space in startups, and lots of mergers that are occurring to be able to take these ideas from the preclinical space into the clinic. And so at the end of the day, there's tremendous promise with gene therapy. There's still tremendous hurdles that need to be usurped if we're going to actually be able to deliver efficacious and safe therapies in clinic. And ultimately, at the end of the day, despite all the finances, I think the needs of the patient come first. And so we do have a long way to go, I think, before we see cardiomyopathies being treated routinely in our clinical space with gene therapy. But I think the road to getting there is actually ahead of us and visible, and we can see these challenges. And I think in 5 to 10 years, we're going to be there seeing cardiomyopathies more and more being treated by gene therapies. And so my take-home points for our time together, improved AAV transduction efficiency, cargo delivery, and CRISPR gene editing, among other advances, have driven a real resurgence in gene therapy exploration for a variety of diseases. There's very exciting preclinical data that we can translate some of these known mechanisms of disease and these new advances into pediatric and adult cardiomyopathic conditions. There are a handful of cardiomyopathy trials that are getting off the ground. Nearly all are focused in adults, and as a pediatrician, I'd love to see more with children. And I think overall clinical trial work and more basic science will lead us ahead to realize the full clinical benefit of gene therapy. And so with that, I'd like to thank you for your attention. Welcome any questions now in our Q&A. Thank you all very much.
Thank you very much, Dr. Landstrom. And now for our Q&A portion, please post your questions in the Q&A feature on the webinar, and where Dr. Weaver will field those questions for us.
All right. As we're waiting for people to type in questions, I'll start and ask one. Who do you think will be the physician or specialty that will be delivering these therapies once they're approved? Will it be within cardiology or some other group?
I think if you take a step back and see kind of what other specialties have done, it's really been a multidisciplinary approach. I think the specialty that is sort of central is a specialty that cares for the disease. So if we're talking about cardiomyopathies, I think the cardiologist, the adult cardiologist, the pediatric cardiologist has to be central to the team that delivers gene therapy clinically. I think they're going to be central to the clinical trials that get us there. They're going to be central to the initial rollout of the therapies in clinic, which are going to be expensive, time-consuming, and potentially logistically challenging to deliver. And then they're going to be central to the ability to translate these findings more and more, hopefully as more therapies become available and as the price of delivering that therapy drops. But I think the cardiologist can't do this alone. I think this really underscores the value of being in a multidisciplinary care center and for these patients undergoing multidisciplinary care. I think colleagues in medical genetics are absolutely critical here to be able to provide optimal care. I think it highlights the needs for genetic counselors, specialized nurses who understand clinical genetics, as well as all the other individuals that go into delivering advanced therapies safely. So whether that's the inpatient monitoring unit or whatever is the case for your particular institution. Overall, I really think it's going to be a team effort. And I think everybody is going to have to really work together to identify the correct patient, to identify the right gene therapy for that patient, to deliver it, and then make sure that the therapy is giving them all the efficacy and should there be an adverse event, to be able to spot it quickly and to treat it.
Thank you. I agree with that emphasis on the multidisciplinary care. I think that will be really important. All right, so one question from the chat. How much do we know about immune-induced myocarditis in gene therapy? Is it primarily the delivery vector, virus, or RNA, or are there patient-specific factors that predispose to this adverse effect?
I love the question. So this is multifactorial. So at its core, it's likely due to the immune system responding to something in the virus. This has led to the kind of development of those sort of lipid nanoparticles that we were talking about, which sort of take the virus out of the equation, because viruses, even the most sort of stripped down new capsids, do have the ability to trigger the immune system. So oftentimes that's sort of really key to that sort of myocarditis type picture. There are a lot of different efforts to usurp that. We talked about sort of lipid nanoparticles to do that. There are some individuals who are looking at really sort of we call it sort of immune naive capsids where the human immune system has never seen this sort of capsid before, thus potentially may not actually mount an immune response. So there's lots of interest there. And there's also lots of interest in leaving the virus out of it and looking at lipid nanoparticles as one other means of doing it. The other aspect of this, and the reason I like this question so much, is there's another wrinkle to sort of participating in early phase clinical trials that are all virally delivered. And that is, if you receive, say, a phase 1 clinical trial AAV9, if you didn't have the immune system recognize that virus going in, it may recognize it by the end of the trial. And that may actually preclude you from taking part in future studies because now your immune system has seen that particular virus, has generated antibodies, neutralizing antibodies against that virus, and thus you may not be able to, say, take advantage of another clinical trial down the road or a bona fide gene therapy if it uses a similar virus. So these kinds of wrinkles, I think, are really important, especially when you're counseling families about gene therapy, whether they may want to take part in it, and this really highlights the complexity of the decision.
All right. Our next question. Safety issues are a real concern to the patient community. What types of questions should families ask their physicians and be aware of in their evaluation to participate in a clinical trial?
These questions come up all the time, and I think it's wonderful. I think if you take a step back, the first question is, do I know my gene mutation? So if you have cardiomyopathy, say hypertrophic cardiomyopathy, do I know my gene mutation? It starts there. And so if your cardiologist has not gotten diagnostic gene testing for your disease, it would start there. Understanding your type of mutation, because without understanding that, really you don't know whether you might be eligible for some of these trials. And also, I would say that understanding your mutation is actually really important for your overall clinical care, including the care of your family. So multiple reasons to know your gene mutation. Second, I would just start asking, what that provider's experience has been in clinical trials or whether they're aware of any. The field is moving very, very quickly. There's lots of clinical trials that are springing up, particularly in this space. And you could tell kind of from the whirlwind tour that there are a number of trials in multiple different places. Just ask about what their familiarity is and realize that perhaps you may need to speak to a specialist in the field if that primary cardiologist is simply not aware. There are lots of opportunities to sort of find this information out on your own through the website for these trials. However, having that trusted interface that you can talk to, ask questions of, is really important. And if that's not your primary cardiologist, I would find a cardiologist that can answer those questions. Oftentimes, the next step then is to just explore the possibility. And many clinical trials have people that are sort of patient advocates who are not directly related with the trial, but their job is to answer questions, give you a thumbnail sketch of what exploring being an early phase participant would mean for you and your family so that you can be well informed. These phase 1 trials are not simple. It's not like you come in, you get an IV, and then you go home. Phase 1 trials are all about safety. And so you can think of it like the safety profile needs to be established, and they establish it in the first patients who get the virus. So if it's a phase 1 trial, you can expect a lot of attention given to safety, and that may mean hospitalization for a number of days. That may mean lots of blood work for days to weeks to years after. That may mean testing. That may mean lots of questionnaires. Sometimes families relocate to one of these sites. It can be a very big deal for families. However, if you're interested, there are lots of resources available. I would start with knowing your mutation and having a conversation with your provider.
All right, so we have 2 questions about where these trials are happening, primarily in the US or overseas, which centers in the world are treating cardiomyopathy with gene therapy, and then are there regulatory factors that make one preferable over the other? That's the US versus overseas, yeah.
Wonderful question. Most of the trials that I discussed today are enrolling patients in the US. There are clinical trials for cardiomyopathies that are enrolling in Europe and some in Asia, although they tend to be a bit more rare. Most of the companies are enrolling in the US. In terms of regulatory oversight, I don't know if I'm the best person to actually answer that. I can tell you from a patient and a care perspective, if you're exploring a phase 1 trial, like I said, it's usually not just going in for an IV and then coming out. Usually it is an involved process where you can expect to be in that hospital for some time, again, as they evaluate the safety of that gene therapy. And so that may have practical implications for you as a family if it's, say, in the US versus outside of the US. I do think that most companies are targeting the US for enrollment sites, but certainly I think people would like to expand into Europe and into Asia because obviously the US is not the only location where there are people who could benefit from these trials. I talked to at least a few families who are of the opinion, like, let's see how the initial phase 1 trials go, and then maybe think about a phase 2 trial, which typically have broader enrollment at more sites. And for the studies that we went through today, that phase 2 hasn't been determined yet. So stay tuned. A good resource for you, for your families, and for providers is clinicaltrials.gov. It actually is a great way to keep track of all these trials. If you haven't been there, you can just search by cardiomyopathy, see who's doing what, who the primary funder is of the trial, where they're enrolling, and the eligibility criteria, all right there, clinicaltrials.org or .gov.
All right. So now we have a couple of questions about that the current gene therapy trials are addressing advanced disease, and how early do you think gene therapy has potential of one day treating? And then sort of along those same lines, are there any gene therapies in the pipeline to prevent congenital heart disease?
Wonderful questions. So I think you're absolutely right. Most of the trials now are targeting the more advanced disease. And I think the reason being is, when you have not established safety, you want to make sure that you are giving a new therapy for a very clear reason. And so a lot of these trials are individuals who had more advanced stage disease, who might have symptoms, who might have disease manifestations that are refractory to standard of care therapy. And gene therapy is sort of coming in as a adjuvant therapy to help with that. But you're absolutely right. I think hopefully as these trials are proven safe, the indications for that gene therapy will undoubtedly expand earlier and earlier. I think there's probably a sweet spot, a Goldilocks phenomenon, where you want individuals who are not super sick to get the gene therapy, and not individuals who are very, very healthy to get the gene therapy, but people who are starting to notice the symptoms and have a good reason to take part in the study, but not necessarily be very, very sick or really quite healthy. Going forward, I would love to see the day where gene therapy could be considered a preventive strategy. For example, I think we've all had that patient with arrhythmic cardiomyopathy with, say, a PKP2 mutation. I'm a pediatrician, so sometimes we see these patients when they're young. Sometimes they're infants. PKP2 variant, cascade identified in the family from maybe a sick father. And so you're looking at a 3-month-old with a PKP2 variant, and our current guidance says, guess what? That infant should never do sports or never exercise. And that is really unsatisfying and just really not acceptable for that family. So I would love to see the day where that child could get a gene therapy, prevent their disease, and they could live a normal, happy, healthy life. We are not there yet, but that's my dream, and I hope we can get there. Oh, and there was congenital heart disease, right? I think the last part of that question. Mm-hmm. My goodness. So congenital heart disease, I think, would be the holy grail of gene therapy. Congenital heart disease, in terms of understanding its genetic and cellular underpinnings, I sort of feel like is about 15 years behind cardiomyopathies. We simply do not understand enough about the sort of complex genetic milieu that leads to congenital heart disease. I'm thinking of things like hypoplastic left heart syndrome, tricuspid atresia, functional single ventricle heart disease. The genetics are quite complex. The cellular mechanisms that drive that are actually very complex and probably a mixture of environmental factors as well as genetic and cellular factors. And so to be able to uncouple all of that and to find a genetic or molecular linchpin that could be treated early on as the heart's developing, I think is quite challenging to see a way forward right now. But I know that there are phenomenal scientists working in this space. And I would love to see that someday. I think it'll take some time first to define those mechanisms, then to show that you can actually perturb those mechanisms early on. And then, potentially you could have a conversation about how you might deliver that perturbing thing, whether it's a small molecule or a gene therapy fetally. But that would be the path forward. But still a ways to go there for sure.
Given the challenges or that risk of that immunoreaction with the phase 1 trial participation, that could prevent somebody from participating in future trials or approved therapies, how would you counsel a patient or family with decision-making about that?
Great question. I think, all of us are sort of used to having in-depth conversations of risk. Oftentimes when that risk is sort of hard to quantify, I fall back on a very simple shared decision-making model. Recognizing that oftentimes this conversation is limited by my own knowledge. And oftentimes, if they want to have a more thorough conversation, they need that patient advocate on the trial side to really spell things out for them. But basically, I try to resist the urge to impart whatever my goals and my preferences are on the family. I try to engage with them as best I can about what the risks, the benefits, and alternatives are of considering a gene therapy trial, and then engage with them and ask, what are their goals? What do they want? And it's really eye-opening when you ask families, like, what are your goals if you were to take part in a clinical trial? You get a lot of information from them, and it may help you to reframe your own thinking on how best to counsel them. And we have a conversation. And the outcome of that conversation may be just, okay, we're going to think about it more, or maybe we want to reach out to that patient advocate and have a more detailed conversation. But a lot of listening, a lot of shared decision-making, recognizing where one's knowledge is limited, and then trying to be as humble as possible about it, because there's a lot we don't know. And I think families are incredibly receptive to that. And I'm consistently impressed by, their ability to process the information and make very reasonable decisions for them and their families.
All right, we have a question from somebody who is early career who's interested in getting involved in this field. Any suggestions for how somebody gets involved and participates in this exciting science?
Yeah, I mean, I think there's lots of ways for trainees to sort of dive in. There is tons of opportunity in the preclinical trial space, and so that's sort of the area of basic science and translational science. There is amazing opportunity to try to find the next best gene therapy target. And we talked about some of the advances that make that possible: gene sequencing, great models like iPSC-derived cardiomyocytes. There are unparalleled opportunities now to identify the next target for gene therapy. If one can identify a new mechanism of disease, a new gene, a new way to manipulate a disease process, to rescue a disease process, tons of opportunity. And so if your interests lie there, just a matter of finding a lab or finding an investigator that can stoke those interests in you. There's lots of opportunity there. In the sort of early trial space, tons of opportunity for learners, for clinical trainees to know how to run a clinical trial. I don't think you necessarily need to be exposed to a phase 1 gene therapy cardiomyopathy clinical trial, but really knowing how do clinical trials work, what sort of centers oftentimes are the home for those early phase clinical trials and why, what sort of patient populations that are selected and why. All those things I think are great areas for trainees to explore and can be done in any sort of larger academic center with an active clinical trial pipeline. Great opportunities there. And then there's opportunities for folks that may not seem as apparent. There's, I think, a lot of focus being given to sort of equity of clinical trial participation. So you can imagine how individuals who might be overlooked and have trouble accessing medical care are often overlooked by clinical trials. And so there's actually great work being done on the implementation science side of things, of how to run a clinical trial that is equitable, that engages the entire community for which the trial wants to serve. And how do you do that? How do you actually engage underrepresented communities in a way that makes the trials better and ensures equitable distribution of whatever the trial shows? Really great work being done there in implementation science, which we need more trainees for. So I think lots of opportunities, and would love to see more people jump into the field.
All right. Next question is whether a patient who is treated with gene therapy can transmit the defective gene still.
This is really important. So there was a few years back, you might remember a sort of a scandal in the sort of gene editing space where somebody, a Chinese investigator, made a mutation in essentially a germline of an embryo, so that they made a genomic DNA change, and then every cell in that resultant embryo and the resultant child carried that genetic change. And it was a big deal because that genetic change was conferred into that child's germline, into the sperm or the eggs of the individual. The reason that's a big deal is because if you misstep, if you change the genome elsewhere, you essentially have created a lineage of individuals who have a gene mutation of, the field's collective making. And all of that sort of really put a moratorium on germline gene editing. And so all the trials that I talked to you about today, all the preclinical stuff, none of that is germline gene editing. That's out. The individuals who are getting gene editing are not getting germline. They're getting it delivered directly to a tissue that's defective. And so I mentioned Terry Hogan in the N of 1 Duchenne's muscular dystrophy trial. That was gene editing just for the skeletal muscle. This is where things like cancer, for example, leukemias, lymphomas, or immune deficiencies actually have the ability to do ex vivo gene editing, where the cells are actually taken out of the body, edited ex vivo in the lab, and then reintroduced into the host. That precludes the ability of that gene edit to happen at the germline. And so that's where the current field is. And I do not see the day, at least in the short to medium term, because we have a lot of ethical things to unpack with it. I don't see the day where we're going to be talking about sort of germline transmission. And I'll also say that one question that occasionally arises from patients is, say I get a gene therapy for my PKP2 variant, can I still transmit that variant to my kids? And the answer is absolutely, because that PKP2 replacement is only replacing the wild type gene in the heart. It's not changing the presence of that gene, which often occurs heterozygously in the germline, in the sperm or the eggs of that person. And then because it's an autosomal dominant mode of inheritance, would be a 50-50 chance of any biological child receiving that variant. Whether or not you've had gene therapy of the heart, that doesn't change.
All right. Let's see. Our next question. Can a patient treated with gene therapy shed virus that can result in family members or caregivers having an unintended exposure and develop antibodies preventing they themselves from getting gene therapy?
Good question. These viruses are classically non-infective, and thus they can't replicate inside the human cells and cause us to be sick, and thus cause us to shed the virus. So in theory, these viruses, once they're given to the individual, don't leave the individual, at least in the infective way that we're thinking of, like, say, maybe a cold can be spread from family member to family member. So that would not necessarily be possible. Although, we are exposed to tons and tons of viruses all the time. And so even if maybe you can't, catch the gene therapy virus from maybe a partner, who knows what you've already been exposed to in terms of viruses, just being a human that walks around our community. And so the presence of neutralizing antibodies in the population for these viruses is high. And really, there's not much we can do about it. We actually want to have neutralizing antibodies against viruses, right? That's a cornerstone of our immune system. But really, there's no way of predicting who might have those neutralizing antibodies and who might not. And I would argue that I don't think necessarily we don't want to have any neutralizing antibodies. I think that would be something where there would be immune consequences because of that. So hard to say, but probably not from the gene therapy, but potentially just from being in the community.
All right. Well, it looks like our last question currently is somewhat of a specific case example question, which hopefully is okay. So it is an 18-year-old patient with ARVC due to a PKP2 mutation, has had several complications, a sudden cardiac arrest, has an IDC, and had 2 electrical storms, as well as had a recent ablation. He's on amiodarone. Should this person be offered, I guess, referral to a gene therapy trial?
My goodness, that does sound like a very challenging individual, and I'm sorry to hear about that case. So if you look at the gene therapy trials that are enrolling patients for PKP2, it would sound like that individual would meet eligibility criteria, 18 years or older, ICD in place, sounds like frequent and refractory arrhythmias. Oftentimes the arrhythmia burden is what they're looking at in terms of whether the virus might be delivering efficacious gene therapy. And certainly they would be PKP2 positive for a loss of function variant, it would seem. So if that's the case, they would seem to meet eligibility criteria. Now, of course, that's a separate question than whether enrollment in an early phase clinical trial is right for that family, which of course is a individualized decision, and that shared decision-making really comes into play there. But they would seem to be eligible and could pursue it.
All right, I think that was the last question in the box and puts us right up with 4 minutes to spare, so pretty good
Wonderful.
timing.
So let me just thank both of you, Dr. Landstrom and Dr. Weaver, for your time and your expertise today. And on behalf of both Hart University and the SADS Foundation, I want to thank all of our participants for joining us today. So have a good rest of the day.